Hi guys, my name is Hannah and today I'll be presenting my scientific communication assessment. I hope you find this presentation informative and interesting. Okay, gamma aminobutyric acid or GABA, G-A-B-A, is what I'm going to talk to you about in this slide and the next few slides. I'm going to talk about how GABA exerts its physiological effect and how it affects neuron excitability in the central nervous system. So GABA is a principal inhibitory neurotransmitter in the vertebrate central nervous system. And activation of ionotropic and metabotropic GABA receptors by GABA reduces the excitability of the cell, which inhibits neural firing. Plasma membrane GABA transporters maintain the extracellular GABA concentration in the vicinity of the synapse and consequently control the activity of GABA receptors. Okay, so GABA is synthesized from glutamate and it gets degraded into succinic acid. GABA receptors are further subdivided into GABA A ion channel receptors and GABA B, which are G protein coupled receptors. I've just put a little diagram in here just to show the GABA going through the neuron and into the synapse and into the postsynaptic neuron. The majority of GABA receptors are GABA A receptors, and they act as chloride channels, exercising large inhibitory effects. And many drugs act on GABA pathways. And that further leads us into what I'm going to be talking about next, and that's how the effects of Valium and ethanol and barbiturates work on that same pathway, which is sort of explained in this diagram here, where you can see the ethanol and the barbiturates and the benzodiazepines going through that same chloride channel. Okay, so now I'm going to talk about the physiological effects of Valium and alcohol and barbiturates. Okay, so Valium is a central nervous system depressant, which works on the same brain system as alcohol and barbiturate drugs. Alcohol and Valium act on GABA receptors in the brain, which lead to similar effects. People who have seizure disorders, anxiety, or insomnia often have less GABA available to moderate the communication speed between neurons, so their neurons fire faster. This can lead to feeling tension, fear, etc., etc. Both alcohol and benzodiazepines, along with barbiturates or sleep aids, bind to GABA sites, allowing the neurotransmitter to remain available in the brain for longer. This allows the neurons to fire slower. For people with the disorders that I mentioned, epilepsy, anxiety, or insomnia, this eases these disorders. So taking both alcohol and Valium or barbiturates together is very dangerous because they both increase the bioavailability of both substances together. They increase each other's bioavailability, which increases the risk of overdosing. And I've just put in a picture here that just shows the GABA channels and how the ethanol and the benzodiazepines work on those same channels that go through with the chloride as well. Okay, so chloride and skeletal muscle. So the type of channel responsible for chloride movement in skeletal muscle is CLCN1 or CLC-1. The voltage-dependent CLC1 chloride channel belongs to the CLC channel transporter family. It comprises of two individual pores which can operate independently or simultaneously according to two gating modes, the fast and slow gate of the channel. CLC1 is preferably expressed in the skeletal muscle fibers, where the presence of an efficient chloride homeostasis is crucial for the correct membrane repolarization and propagation of the action potential. Okay, so channelopathies is a disease caused by dysfunction of ion channels. Mutations in the CLCN1 gene cause dominant and recessive forms of myotonia congenita, or MC, a rare skeletal muscle channelopathy caused by abnormal membrane excitation and clinically characterized as muscle stiffness. So just a reference list here for all the information that I have used. Everything that is spoken verbally throughout the presentation is cited on each slide. Also, all pictures used are referenced on each slide. Okay, so thank you very much for listening, and I hope it has been informative. Thank you.